pleasure to be here. I've missed these Friday seminars. They're always very nice. Yeah, message says the recording has started, so let me see about sharing my screen and getting the slides up. There we go. I think that's worked. If it hasn't, then somebody needs to mention in the chat or shout at me. I'm going to assume that you can see my screen now. And if we go to the presentation view, now I have no view whatsoever of the Teams chat. So I'm really at the mercy of somebody telling me that there's a question there if there is a question, but I'm very happy to take them throughout the talk. I've never delivered a talk like this online, so I've written this talk to be roughly of the length that I would normally give in an in-person 50-minute to 1 hour seminar, but I have no idea what the online format will do to the length, so we'll find out together. Anyway, this talk is basically an extended example almost, in that I wanted to see how some of there's over the past few years been a number of these kinds of new non-reversible MCMC algorithms which purport to have some very nice mixing properties, and I'm interested in a very particular inference problem to do with coalescent models, which are standard models in population genetics, and so that's a setting in which standard Metropolis type methods have mixed notoriously poorly, and this is my attempt to understand whether and to what extent these non-reversible methods might be able to help with that. The short spoiler is that, as far as I can tell, there's no one definitive answer, but there are definitely cases where it looks like they can be helpful. Here's an outline of what I'm going to be talking about. So I'm going to briefly introduce and define the coalescent process. You don't need to have any understanding of what it is or how it appears in PopGen. Then do a one-slide introduction to Metropolis-Hastings, which always is probably unnecessary in these seminars, but particularly if it's being recorded, that hopefully may be helpful. And then talk about what needs to be done to the coalescent in order to write down a zig-zag algorithm for it. So zig-zag is one of these non-reversible processes that has grown to prominence over the past few years, and that's the one that I've chosen to work with, largely because my office in normal circumstances is across the corridor from Gareth Roberts, and so I can ask questions very easily. And yeah, then the second half or so of the talk will be some numerical results for simple examples, which I've had sufficient time to code up. These are nowhere close to practical examples of real data set analyses in genetics these days. These are really just toy problems, but I'm comparing zigzag results to corresponding relatively simple Metropolis algorithms and trying to get an idea of whether it's worth trying to extend this to more involved problems. So without further ado, the coalescent process. So this is a gold standard model in population genetics, and we're thinking of genealogies of finite samples of individuals. So if you put yourself in the mindset of a biologist, you have an organism that you're interested in, you go out and sequence a bunch of individuals and you get their DNA sequence data. And what you would really like to have is not only all of their DNA sequence data, but also all the sequences of all of their common ancestors, all the way up to when there is only one common ancestor for your whole sample. And if you had all of that, then roughly speaking, you could solve PopGen on pen and paper. It's a caricature, but it's not, while it's not misleading. Life would be much simpler if you could have all of that. But of course we can't. There's some instances of ancient DNA and things like that being available now, so you can have partial information of some of the sequences of the common ancestors maybe, but in virtually all practical settings, that sort of thing is not available. So instead, what you do is data assimilation. You propose a model for the common ancestry and try and integrate over the model using some sort of MCMC method typically. And the coalescent is the gold standard model for various reasons, which I won't go into very much, except to say that it's robust in the sense that it doesn't matter too much what sort of precise assumptions you make about what how you model the population evolution. The resulting process resembles the coalescent for a large class of models. So what is the coalescent? It's convenient to think of it in terms of a partition valued stochastic process. So little n is a sample size, and the initial state of the coalescent is this trivial singleton partition where every individual corresponds to its own partition block. You have holding times, which have exponential distributions. All the holding times are independent. So essentially what's happening is each pair of blocks is trying to merge at rate 1. And yeah, when one of these clocks rings, 2 randomly chosen blocks merge. And you indicate that in the state space of the process by merging the 2 blocks of the partition. So at later times, 2 indices I and j belong to the same block, if and only if they have a common ancestor by that time. And that gives you the notion of a kind of prior distribution on genealogies. So you can think of the coalescent as a prior for the missing data of the genealogy. In order to connect it with a likelihood, you need to put mutations on it. That's, after all, what you're actually observing through DNA sequence data. I'll give several interpretations of what mutations might mean throughout the talk, 2 of them to be specific, but the overall theme is that there's some parametric mutation rate, thetan 2, and mutations occur on the branches of the realized tree as points of a Poisson process with this rate thetan 2. The division by 2 is purely aesthetic. It simplifies some formulae in some cases, but of course it's superficial. So this is what a possible realization might look like. You have 5 lineages here, these horizontal lines on the right-hand side of the screen, and they have some mutations. And then the remainder of the screen, the sort of tree structure on the left, is a possible realization of the coalescent giving rise to this observed sequence of mutations. So first I want to say simulated, but in fact I mean drawn these branches of the tree, and then overlaid some mutation points along it. I should emphasize that the vertical separation between these lineages means nothing at all. It's just the horizontal branch lengths that have any meaning. The vertical ordering of the samples has been chosen in such a way as to make the coalescent easy to draw and easy to see, but any reordering of the rows of this thing would result in the same object. So this then is the sort of missing data problem that we're faced with. The biologist can't see the coalescent tree. It doesn't have, they don't have ancestral information for their samples. Instead, they just have the sampled points, these 5 DNA sequences on the right-hand side, and the task is to integrate over all possible coalescent trees that could have given rise to this data. So that's this notation on the bottom. Probability of data given parameter is probability of data given ancestry and parameter times probability of ancestry and parameter. And so this is a horrible integral over a space of trees, which it won't be surprising to anyone to hear that you can't make any progress on this, even in simple cases. Well, in very few simple cases, you can make progress on this analytically, and numerical methods can, again, in simple cases, take you up to samples of size 50 or so maybe, but beyond those sorts of numbers, MCMC is basically the only game in town for approximating these integrals. So this is the single slide primer on how a Metropolis algorithm operates. Forgetting for the moment about trees or anything like that, and just a generic algorithm. Suppose we have some target density pi that we want to simulate from. We start with an initial condition. Doesn't really matter what it is, and iterate through time by sampling a proposed perturbation of your current state. So y you can think of as a perturbation of xi minus 1, q is the transition density giving rise to the perturbation, and then you have this accept reject correction. So the simplest thing to visualize is to take q to be symmetric, in which case it cancels from the acceptance probability, and you sort of see that if your proposed point is of a higher probability under your target distribution than your current point, then you always accept. So you do hill climbing, and If it's of a lower probability, then you still accept with some probability. So that balances exploration and exploitation, and turns out to ensure that the Markov chain formed by this mechanism has pi as its unique invariant distribution under very mild conditions. And you can imagine doing this on sort of more complicated geometrical objects fairly easily as well. So I've spoken about target density pi here, but if your target is a distribution defined on spaces of trees, for instance, then Q could easily represent some sort of local rearrangement of a tree, and all of the quantities involved in here are still easy to give meaning to and relatively straightforward to code up in simple cases. So the Metropolis algorithm is nice to apply to the Coalescent model precisely because it has this kind of robustness to it, in the sense that the space over which your target distribution is defined can be really quite irregular before it gives you any conceptual problems with the algorithm. That said, it does not work particularly well for coalescent processes. Several different kinds of algorithms have been tried, and they tend to scale very, very poorly. Here's a woefully incomplete list of reasons for that. So coalescent targets are defined over spaces which have discrete components in the sort of natural way to view the space, which means a lot of the tricks that you can use in terms of more sophisticated algorithms to accelerate mixing are not easily available. Something like a Mala using a Langevin diffusion, you need to be able to evaluate gradients of your target, and you don't even know what that means. Conditional distributions tend to be intractable, so it's not easy to define something like a Gibbs sampler. There's heavy correlation between the tree and the parameter, so taking the parameter to be the mutation rate from the previous slide, a given data set might be equally well explained by a high mutation rate and a short tree, or a long tree and a low mutation rate, and it's difficult to get MCMC algorithms to mix well between those two. Targets tend to be multimodal. It's quite often the case that the same data set can be roughly equally well explained by various different tree topologies. The dimension of the space of binary trees with n leaves grows fantastically quickly with the number of leaves. It's at least factorial. There's an expression later on in the talk for something to do with that. And also, you don't even really know what good mixing should look like, because things like optimal scaling results don't tend to be available. For instance, if I flick back a couple of slides, you've got these exponential holding times given on this slide, and they operate on wildly different scales. You can see if you have the first holding time is of order 1 on n squared on average, where n is the sample size, and the final holding time is always of order 1. So you can sort of you can look at rescaled versions of the algorithm where you try to harmonize those scales, but ultimately you're still looking at a process which does not consist nicely of components that live on a comparable scale as the sample size grows. So that's one of the major motivations for this work, is not to really address any of those technical difficulties, but to find a nice way to implement some more sophisticated and potentially faster mixing algorithms for coalescence. There's a reference to a paper later on in the talk where that's been done for a Hamiltonian Monte Carlo algorithm. So I'm not the first one to try out something like this. And both the work I'm presenting now and the Hamiltonian algorithm rely on somehow embedding the discrete space of binary trees into a continuous space. My work uses an embedding by Gavriushkin and Ramond, which ends up being quite nice and natural. The earlier HMC work uses a different embedding, which I don't think works so well in the coalescent setting, but I'll comment on that in more detail at the end of the talk. For now, let me just introduce this Gavriushkin-Ramond embedding, which is referred to as tau space, and this will be the state space on which my algorithm operates. So we are thinking of binary trees, and we want to encode each tree as this tuple EN-TN. The TNs represent times between successive mergers. So T1 is just the time between the leaves and the first merger, T2, time between the first merger and the second merger, and so on and so forth. The EN is a bit more implicitly defined. It's called the ranked topology. So it's basically a labeling which tells you which two nodes merge at each merger event and the ordering of all of the merger events. So you can imagine labeling your leaves with the natural numbers 1 to little n in the same way as we did for the state space of the coalescent, and then introducing some labeling of the ancestral nodes that arise out of merger events, which indicates which leaves participate in each merger event, something like that. So the tau space then is built out of taking one copy of the space of possible intermerger times for each possible ranked topology. So there are fantastically many possible ranked topologies for a binary tree with n leaves, and for each of those you take a copy of this space of intermerger times, and you attach them together by gluing common faces of these what are called orthents, these copies of Rn plus 1. So this thing there is referred to as an orthent. And by gluing common faces together, you can take this whole mess of absurdly many copies of this space, and that forms one connected space. You can see here is the number of these ENs is ridiculously large, but in principle this still works. You get one huge space, but it's connected and finite, and not finite, connected and locally finite, and has sort of, except for at these orthent faces, it even looks just like Rn. So here's a diagram which I've shamelessly stolen from Gavriushkin and Ramond, where they have the tau space with 4 leaves, and they've drawn 1/3 of it. So you would need, what's this got? This has got 1, 2, 3, 4, 5, 6 orthents on it. You would need another 12 in order to complete the whole space. But you can sort of see, for instance, if you look at the bottom, I don't know actually, can you see my mouse? Richard, can you shout out and tell me whether you can see what my mouse is pointing at on my screen, or if I need to draw on it?
No, I can't see your mouse, I'm afraid.
All right, fine. So if I look at this orthent here, then that's one particular ranked topology of mergers. So 3 and 4 are the first nodes to merge, then the common ancestor of those merges with 2, and then the common ancestor of that merges with 1. And if I trace across this boundary over here, what essentially happens is the edge connecting node 2 to its ancestor shifts up along here and back down this way, and that's a continuous transition across that edge. And in fact, this edge over here is adjacent to exactly 3 of the orthents. corresponding to 3 different ways in which you can resolve there's a kind of degenerate topology that fails to be binary right at this edge over here, which, let me see if I can tax my drawing ability and no, I probably shouldn't have been brave enough to do this live. It would be the tree that looks like that, which lives exactly on that edge. And there's the different orthents to which it connects correspond to the different ways of resolving this triple merger at the top. Okay, so this is a reasonably nice space. It's sort of conceptually straightforward to see how one might implement things like piecewise deterministic Markov paths here. So that said, let me tell you what the zigzag process looks like. So this is still a completely general formulation again. I don't think this is the first paper in which something like this appears, but it's one of the main ones, this Bjerkens, Fernhead, and Roberts at the bottom. So let's think again for a moment of a density on Rn that we wish to target, and we augment the state space with velocities. So each coordinate within my state space gets associated with it a velocity, which I'll take to be either +1 or -1. And I start from some initial position again, and now evolving continuous time, and I do that as follows. So I have these rates, these lambda i's on the bottom, and they correspond to flip rates. And when a flip happens at a particular coordinate, that just means that the velocity flips. And in between these flips, the algorithm just proceeds by tracing a continuous straight line with whatever the current state of the velocity is. So this is somewhat implicit notation here, but that's kind of the meaning of this, is we sample a waiting time y, which is exponential with the total of the rates. So the rates are, of course, position dependent, and progress the algorithm up until the waiting time by following this straight line path just given by the velocities. Then once the clock has rung, we sample from this categorical distribution which velocity actually flips and flip that velocity and carry on. And again, this form of the velocities has been chosen in such a way to ensure that pi is the unique invariant distribution of this algorithm. So okay, that's the zigzag process on Rn. Here's what a simple example looks like. This is not my simulation. This is from Bjerkens, Fernhead, and Roberts of a 2-dimensional target with these kinds of S-shaped contours, and you can see that there's 90-degree turns corresponding to velocity flips, and between those the algorithm just proceeds on smooth straight lines. So how does one go about implementing this for a coalescent? Well, it's sort of straightforward to see what happens within each orthent. So as long as you don't touch one of the boundaries, then everything's okay. So let me first talk about what the actual coalescent posterior looks like. And it's sufficient to keep in mind a single orthent for now, and we'll deal with what happens at the boundary points in just a moment. So we can regard the rank topology just as a set of edges of the coalescent tree, and we have little n observed sequences, and here's just some notation that makes writing down formulas a little bit easier. So m of gamma will denote the number of mutations on gamma, and we say that ti is included in gamma if gamma spans the ith interval. So remember that ti is the amount of time between the ith and I+1th merger. And so a given edge may or may not be involved in that merger, and so any particular edge its length may be a sum of multiples of these waiting times, depending on which merger it was born in and in which merger it terminates. And L gamma will just be the length of an edge, which is given by the sum of all of the time intervals which it spans. I've written M gamma for the number of mutations on a branch. I'm implicitly assuming here what's called the infinite sites model of mutation. So let me engage in bad practice and flick back through my presentation. So essentially what the model assumes is precisely what's depicted in this diagram. So each individual is actually a continuous interval of DNA. So you can think of this being just the unit interval 0 to 1. And each mutation that arises on the coalescent tree has a location which is sampled uniformly along the interval. And then mutations are inherited down the branches of the tree. So it could be that this mutation has location X, which would be this point over here. And this mutation has location Z, which would make it this middle mutation over here, and so on and so forth. And so this makes it possible to determine for a given tree and a given data set precisely how many mutations must have arisen on each given branch, which is why that M gamma is a well-defined concept under this model of mutation. Right, given all of that, the full posterior target on ancestral trees, if you will, is given on the bottom of this slide. So this first term over here is basically a Poisson kernel, which encodes the fact that given branch lengths, mutations have conditionally Poisson distributions. There's an e to the minus theta on 2 term missing, which is actually over here, which completes the specification of the Poisson kernel. And the rest of this second line, except for that theta, is just the coalescent prior, the squared, the independent exponential waiting times. If I wanted to be proper and Bayesian about this, then I would include here a prior on the mutation rate. But I've taken that to be improper throughout this talk, so uniform on the whole positive half line. If you took a proper uninformative prior, then it would change basically nothing throughout the talk. Under this model, you can write down what the flip rates have to be. It's a little bit laborious, but it can be done. And they look like this. So I'm not intending to for everyone to fully internalize the full formulas. I don't think they're especially instructive. Just to say that they involve sort of not necessarily order n summations. So these are not especially cheap to evaluate, but all of these terms that appear are composed out of finite sums, and you can evaluate these with finite computation without too much difficulty. You can also get nice expressions for dominating rates. So why does one need dominating rates? Well, these answer the question of how do you actually simulate these times of flip events in practice? Because if I flick back through to my slide on the zig-zag algorithm again, we have here this expression, which involves an integral over lambdas. So somehow you need to be able to simulate events from this exponential distribution, and the usual trick for how one does that is you come up with a dominating rate, which is not state dependent or is state dependent in some tractable way, simulate from that dominating rate, and then use Poisson thinning. So that's exactly what the aim is to do here. And these lambda stars work as dominating rates. And again, some of these turn out to be very simple. This first one in particular is an order 1 evaluation, but the rest of them are finite sums, but sums which have order n terms in the worst case. They also have integrals that are about as tractable as the sums themselves, so you can actually write down explicitly what the integrated intensities is, which makes simulating those exponential random variables particularly easy because you can just do inversion sampling quite easily. Okay, so the big elephant in the room thus far is that I've glossed over entirely what should be done if my process ever actually hits a boundary of these orthants. And that turns out to be rather simple, and the reason that makes it simple is that this target pi theta is continuous at the boundaries of rank topologies. That's a fortunate property of the model rather than anything deep. So there are bound to be similar sorts of tree models which you could embed into the same space for which this is not true, and then you'd have to do something more complicated. But for this coalescent, continuity of the target means that you can get away with a very simple Metropolis-Hastings argument. So you can think of arrival at a boundary as a Metropolis-Hastings proposal to cross that boundary. And so you, because the rank topology is part of the state space of the algorithm, even when your process sits exactly at the boundary, it remembers, but in a Markovian way, which orthant it derived from. And so you can simply propose one of the other two orthants uniformly at random, apply a Metropolis correction, and continuity means that correction will always be accepted with probability 1. And so actually there's two kinds of boundaries. There's these double points where only two orthants meet, and there you always propose to move to the other orthant and accept with probability 1. And there's triple points where three orthants meet, where you propose your step uniformly and acceptance probability remains 1. This one part is very easy to implement. You hardly have to calculate anything, simply you simply change the orthant, which is the current state space of the algorithm, and ensure that your velocity keeps pushing you away from the boundary in the new orthant. And so this is a sort of this relies on a Metropolis-Hastings argument, but it doesn't really render the process reversible in any meaningful way, because as soon as you arrive on a boundary, you immediately cross it, and your velocity will keep carrying you away from the boundary. So the only way that this introduces the kind of random walk behavior that Metropolis steps are notorious for is if the new orthant you've derived in is actually genuinely very unlikely under the target, in which case you'll tend to flip velocity very quickly and return to the boundary. One thing which is worth mentioning as well is I was indicating earlier that analysis of Metropolis algorithms was made difficult by the fact that these waiting times operate on wildly different timescales, and that's something you need to account for in this zigzag process as well. Namely, if you choose your velocities to all be ±1, like I did when I introduced the zigzag, then your algorithm will in practice perform very, very badly, because a velocity of +1 on a holding time whose typical scale is 1 on n squared for n large will just keep triggering events constantly. And so that single coordinate will mix in very, very well, but all of the other coordinates will barely move at all. And so what I found was that the kind of somewhat obvious trick works where you want the velocities to be inversely proportional to, or rather proportional to, the scale of the component. That's probably not surprising to anybody, but It's nevertheless nice to see confirmation that seems to be the correct thing to do. That balances everything out nicely. Okay, so that gives us a fully specified algorithm. Doesn't really tell us very much about how we should expect it to perform. I don't think there's even an ergodicity proof for this algorithm at this point, actually. There are ergodicity proofs for zigzag algorithms in general, but I'm fairly confident that this coalescent example falls outside the scope of any of them. I saw earlier Gareth was in the chat, so he can correct me afterwards if I'm being uncharitable, but I believe that's the case. But anyway, let's see how it performs on a practical example. So this is a dataset from Ward et al. From 1991. It's rather small and simple, consists of 55 sequences. I think there's 18 mutations there, and this tree on the slide is not a coalescent tree, but it's just a graphical depiction of the relationships among the sequences. There's fewer than 55 leaves, so some of these leaves were observed multiple times, but this anyway is a fair idea of the complexity of the dataset. 55 leaves, 18 mutations. Modern datasets involve millions of leaves and probably millions of mutations, so that gives you some idea of how cutting edge this example is. And yeah, this slide is comparing the mixing of 3 different algorithms on the marginal posterior of the mutation rate. I don't know how visible the titles are on everyone's screen. So the top left is a Metropolis-Hastings algorithm, the top right is the zigzag algorithm I've just described, and the bottom row is a hybrid algorithm of the two. I've not told you what Metropolis algorithm I'm using. It's not any of the standard off-the-shelf ones that are available. Rather one that I wrote myself for the purpose of this comparison. Basically, it's doing subtree prune and regraft moves. For the tree topology, it's doing moves in which all of the branch lengths are perturbed by small Gaussian perturbations, and then it's doing a random walk metropolis on the mutation rate. So it's sort of a basic but functional Metropolis algorithm to try to give a fair comparison to what's kind of a relatively simple and off-the-shelf implementation of zigzag. The Metropolis algorithm, I should say, to the extent that it's possible, has been tuned to give acceptance probabilities of about 1/4 for all of excuse me, each of the various steps. Though, like I indicated earlier, strictly speaking, we don't know that that's the optimal thing to do. But it should be as good a rule of thumb as any. The hybrid algorithm is the continuous time zigzag process, except that it also takes Metropolis steps at a rate which is independent of the state of the process. So it's kind of a combination of the two. And what you notice, or what I notice at least, is that the zigzag and hybrid processes seem to be suffering from visibly lower autocorrelation, but that's largely attributable to the fact that they don't really explore the upper tail of the mutation posterior particularly well. So the Metropolis is finding it much easier to reach very large mutation rates. And that will be a theme throughout all of the various results. Here's a different functional, so instead of looking at the mutation rate, I'm looking at the tree height, which is just the sum of all of the between coalescence times. And this is the other sort of trend that we'll see throughout the talk, where you can really see the poor scaling of the Metropolis algorithm. It's even after a million iterations of the algorithm, this time series is somewhat visibly autocorrelated, and the zigzag and hybrid processes are doing a much better job of mixing and also opposite to the previous slide, doing a much better job of reaching high values of the tree height. A somewhat more quantitative summary. So here I've listed 3 functionals. Theta is the mutation rate that we saw, tree height is the tree height that we saw, branch length is just another measure of the size of the tree. So instead of just being the sum of all of the holding times, it's the sum of all branch lengths in the tree. So some of the various holding times are multiplied by various constants. It's not dissimilar to tree height, and the performance is similar, which is why I've not included scatter plots of it. The top 3 rows are effective sample sizes per second estimated for all 3 of these functionals, as well as runtimes of the corresponding algorithms, and you can sort of see that the zigzag and hybrid processes were an order of magnitude faster and an order of magnitude or 2 have effective sample sizes which are an order of magnitude or 2 higher. The other 2 triples of rows are me trying to get an idea of scaling properties of these algorithms in various ways of complexity of the data set. So theta equals 5.5 ended up being roughly the posterior mode of the mutation rate for this first simulation. So I've fixed that, increased the sample size by a factor of 10, simulated some data, and rerun all 3 algorithms and seen how things change. And the runtimes change roughly similarly. The effective sample sizes of the zigzag and hybrid collapse rather badly, and the Metropolis basically stops working at all. So numbers decorated with stars indicate mixing that was so bad that the effective sample size estimate is completely unreliable. So it should really be interpreted as saying that the Metropolis hasn't worked, or at least that I've not run it for anywhere close to long enough to see output that suggests that it might have worked. The final triple is a different complication, so I keep the sample size as it was at 55, but increase the mutation rate by a factor of 10. So this puts in a lot more of these mutations. And here the zigzag and hybrid algorithms perform really, at least to me, remarkably well. So the performance seems to decay at most linearly in the number of mutations that take place, whereas the Metropolis again really struggles to scale to this scenario. I let it run for 10 times longer than it did in the original problem, but it just produced what's obviously numerical noise. It had not at all mixed across the space yet. And I think what you're seeing here is when I was showing you those somewhat horrible formulae for the flip rates, the fact that the zigzag and hybrid flip rates were linear in the sample size. That's really what I think is driving this process, driving this observation that the Metropolis and hybrid methods scale very well with the number of segregating sites, the number of mutations, which you can kind of think of as a proxy for the sequence length. So how long has the sequence been that has been sequenced? But everything still scales badly in the sample size. The next couple of slides are graphical illustrations of these larger sample size cases. So this is the mutation rate. Again, Metropolis does a better job of exploring tails, but is visibly autocorrelated, whereas the zigzag and hybrid look far more stationary. Tree height, this is really the slide that makes the point, I think, that this Metropolis output is unusable, whereas zigzag and hybrid seem to be performing perfectly nicely. And this is now the mutation rate, the higher mutation rate case. You can see Metropolis struggling and zigzag and hybrid remain stationary, but not particularly good at accessing tails, and tree height, same story. Okay, so that's the case for one coalescent model, but it's a pretty simple one, and in fact it's one for which you would probably not in practice need to do MCMC anyway. If you truly believe that your data is described by just the Kingman coalescent under infinite sites, then you still need some MCMC in your inference pipeline. The target distribution, the posterior, is not tractable, but there are things you can do to substantially simplify the inference problem from just running a naive MCMC. So I wanted to try it out on a more challenging model as well, which is given by the so-called finite sites, finite alleles coalescent. And the only thing that changes with this guy is the interpretation of the mutation events. So I haven't got these continuous intervals as my DNA sequences anymore. Instead, my DNA sequence is given by a single letter, and the mutation rates are just mutation from one letter to another, and again inherited down along the branches of the tree, and a subsequent mutation can overwrite the previous one. And this results in all sorts of phenomena that were not possible under the infinite sites model. So for instance, over here there's a mutation into an A, which is overwritten by a mutation into a T, and then further overwritten into a back mutation back into A again. And under the infinite sites model we looked at previously, all three of these mutations would always be visible in the sample of DNA sequences at the leaves, whereas over here this A sample has lost all track of the fact that there was ever this T mutation that affected it, and in fact a kind of posterior distribution would be far more likely to group the two A's together rather than the A and the T. Likewise, you can get mutations like this, in which the mutation itself is invisible because it's mutating from C to C. And yeah, this I already kind of emphasized, that you can get many independent, the same A mutation can arise many independent times across disparate regions of the tree. So all of these aspects make this a far more challenging model for inference. You need some rather horrible notation in order to describe this, and I guess I've still got a little bit of time left, but I probably won't go through this in too much detail. But essentially the overarching idea is you have a finite alphabet of symbols as well as a finite set of sites, and the tree on the previous slide was of a single site, but in general you think of a DNA sequence as being essentially H to the S in the notation on this slide. So each leaf consists of S sites, and at each site occupies one of these letters, and the tree is common for all sites, which correlates all of the various sites. Conditional on the tree, the sites are independent. P will be a mutation matrix, so it's a matrix which describes when a mutation happens, what's the distribution of the mutant type arising at that site, and we'll assume that it has a unique invariant distribution. Shouldn't have said density here, unique invariant distribution. And finally, this Q is the transition semigroup of the mutation process, so of the compound process, Poisson process, which has jump rate theton 2, and when a jump happens, you sample the resulting jump from stochastic matrix P. And now again, you can write down the corresponding coalescent posterior. You've got here the interarrival times, the density of the interarrival times of the coalescent, and this expression is writing the resulting mutation likelihood, and it's doing it by basically going through this coalescent tree on the previous slide and summing. So we're assuming that we observe the types exactly at the leaves, and we then associate variables corresponding to the types of all the internal nodes, and sum over all of those. So that's what this summation over here is doing. It's summing over all the types at the internal nodes. And then we have conditionally independent Poisson transitions between nodes over here. And this is conditional independence across sites. You can write down the jump rates, the flip rates, I should say. They're even worse than they used to be. I could only fit one of them on the slide. I couldn't fit the one corresponding to the mutation parameter. And this time they are not only linearly scaling in the sample size, but also scale linearly in the number of sites and in the number of possible types at ancestral nodes. So this is substantially worse in practice in terms of scaling. But they can be written down at least in terms of tractable quantities. So you can, if you were to look through and digest this formula, you would see that it basically depends upon these mutation transition probabilities as well as the gradients of the mutation transition probabilities. So these are all quantities which are typically tractable, chiefly because they're only looking at transitions at a single site at a time. However, that rate on the previous slide does not have a nice upper bound. So we got these bounding rates for the infinite sites model. That doesn't work here in general. What does work is time localization. So if you make sure that the time interval you're looking at is such that at most one branch can reach length 0, then you can bound those quantities on the previous slide. That's what this capital T is doing, is it's trying to express a time interval depending on the current state of your process, such that across that time interval, the mutation rate can shrink to no more than half of its current value. So that will localize it away from 0, which would cause divergence in the rates. And also those interholding times with negative velocities can't shrink in such a way that more than one of them can reach 0. The first holding time you have to treat as a slightly separate special case, but that's essentially what's going on here, is this kind of time localization. And that then on this time slice 0 to capital T, you can write down bounding transition rates which are actually independent of the state of the algorithm. And you can also evaluate and sample from all of these in linear time. That was demonstrated in this delightfully named paper by, I think there's 7 authors or so here. Gradients do grow on trees. So this is still linear, although I have to emphasize that the constants are pretty horrible. So again, I'm going to conclude just by a quick demonstration of a simulation study on this model. This is a simulated data set. It was simulated by Griffiths and Tavares in 1994, consists of 50 sequences and only 3 different types of sequences. So 1 and 2 is a 2-point alphabet on each site. There's 20 sites in the row, sample size 50, 13 replicates of the first type, 21 of the second, 16 of the third. And yeah, the upshot is that everything will be a lot slower, but qualitatively the results will be similar to what we saw for the infinite sites model. Metropolis mixes nicely for the mutation rate and is better at exploring the upper tail of the mutation rate than the zigzag and hybrid models. Metropolis does not do particularly well for tree heights. Zigzag does better than that. Over here, I had a slightly higher proportion of Metropolis steps in the hybrid step than I did in the first example, which is why this hybrid picture looks more like Metropolis than zigzag here. But by changing the rate of the Metropolis step between 0 and infinity, you can kind of interpolate between these algorithms at any point you like. Effective sample sizes per second. Well, the real takeaway message here is that this linearity of the flip rates, not only in the sample size, but also in the number of sites and the number of possible internal node type configurations, really kills you. You can see that I'm measuring runtime in minutes rather than seconds, and these numbers are much, much higher. Effective sample size wise, well, the story is more or less the same. Metropolis does well for the mutation rate compared to zigzag and hybrid, but does much worse in the tree height and branch length dimensions, and also starts failing more rapidly in these. So this is the same sort of trick where I've increased the sample size by a factor of 10 and increased the number of sites by a factor of 10, and the sample size kills Metropolis. Actually, this number of sites, all three perform still more or less similarly. So the advantages, such as they are, of this zigzag method in the finite alleles case are far less clear than they are in the infinite sites case. In the infinite sites, it seems pretty clear that apart from this mixing of the mutation rate in the upper tail, it was performing substantially better, but that does not seem to translate well into the finite sites picture, chiefly because the summons, the number of summons you have to evaluate to get the flip rates is so much larger. Here's the same trace plots for these two scaling pictures. So for sample size 500, you see this is a really striking difference in the extent to which the algorithm is able to explore the upper tail of the mutation rate posterior. These both have the same mean. I've checked the estimators and they are identical to within 2 decimal places or so, but the mixing is dramatically different. But then if you look at tree height, Metropolis is clearly worse. And the same when you increase the number of sites. Mutation rate wise, everything still more or less holds up, but with tree height, zigzag starts doing better. This is the final slide, which tries to sort of summarize some of the things that I've highlighted. Zigzag seems to be very well suited to these kind of boundary crossing events which arise when you embed discrete variables into continuous space. So the trick of this embedding trick has been used a number of other times. Reference 9 is the Hamiltonian Monte Carlo algorithm for coalescents, and 10 and 11 are 2 other examples where people have sort of, well, 10 is another example of a similar trick. And the kind of discrete time nature of the numerical integration that you have to do in order to implement an HMC step means that, especially in reference 9, the authors had to work really hard, both analytically and in terms of code, to implement their scheme in the first place and to prove that it works. The boundary crossings were a real pain, judging at least by the fraction of the paper they have to devote to dealing with them, whereas the fact that the zigzag has time continuous paths and very simple behavior along those paths means that these are essentially trivial. For coalescent models specifically, we get dramatic improvements in mixing under the infinite sites model in particular. This third bullet point is trying to address the poor exploration of the mutation rate. I think that has to do with the strong correlation between the tree length and the mutation rate. So in order for the mutation rate to get large, the tree has to get short. And it has to stay short for a long enough amount of time for the mutation rate to increase. And the Metropolis algorithm, with its somewhat non-local update, can jump to a high mutation rate in 1 time step, whereas the zigzag algorithm has to traverse all of the intermediate mutation rates with its continuous path while the tree has to remain short. And if the tree happens to also grow a bit, then the mutation rates progress upwards will be impeded. I don't know whether there's a potential failure mode for ergodicity there even. I really have no idea which way I would guess that to go, but in any case, it does seem to be a practical concern. And yeah, finally, this time localization that I used for the finite sites model, this second part, seems like a nice and general trick to implement this flip mechanism for cases where flip rates are unbounded. And with that, I will stop and thank you for your attention.
Okay, thank you very much. I'll give you a real clap. And if anyone else wants to put comments in the chat now or questions, I should say that if anyone, I'll put my camera on, if anyone does want to ask questions kind of live, then there are more people here than we started with, so I should tell everyone that this is being recorded, and by participating you consent to us keeping the recording and using it for various purposes. Okay, so just before anyone else, so if anyone's got a question, write in the chat. Just before I go get further down, Gareth said thanks for the talk and had some comments which are now on your screen, so everyone can see that. Johannes said thank you. Zia said thank you. So so Dario, do you want to ask this question live so we can see you and see what you so Jere can see your face when he's answering the question to know check whether you understand?
Yeah, I just wrote my question, but yeah. Hi everybody. I was just speculating what can go wrong if one introduces just a mild complication, which is relative to introducing variable population size. So essentially what I remember the slogan being is that when you introduce variable population size in the same model, what happens is that you introduce a stochastic time change to the coalescent tree, but the tree structure remains binary, which is good news. And also, I suppose there are also a lot of explicit formula from which you can calculate quite directly, probably the inter-arrival times, but I was wondering if there is anything computationally that can go wrong.
So you're right that there are explicit formulas. They are, however, only available for the prior. So as soon as you have a data set that you condition on, none of those formulas also become intractable. Let me pull up my talk so I can point to a thing or two as I so let's take the simpler case and look at this posterior expression over here. So what variable population size would do is it would make this term over here more complicated because the distribution of the holding times would become more complex. This mutation likelihood part stay more or less the same. Computationally, what could go wrong? Well, the most important thing is my mental and physical well-being. If I tried to implement one of those things, they are horribly complicated to code, which is why I haven't tried it. But if you had some sort of, say, parametric family of candidate population size trajectories, then there is no conceptual difficulty in putting that extra parameter into the model, giving its own zigzag velocity to those two population size parameters, and the bounds on your flip rates become somewhat more complicated. But at least ensure as long as you ensure that your population size trajectories are bounded above and away from zero, then in principle nothing can go wrong. There's going to be more correlations introduced as well, and that might impede mixing in some regions of parameter space that I don't know, but the same algorithm certainly remains implementable even if you add more parameters.
Thanks.
Okay, so Anthony had a question. Anthony, do you want to ask that live or shall I that out? If you appear in the
I
next?
mean, sure, I can say something. Yeah.
Go for it.
Hi, Jerry. That was really interesting, and I should say that actually Andy, Wang, and Christoph are also listening, but they couldn't connect, so they're tunneling through Zoom. My connection. We were wondering. Well, I mean, me mainly, but I guess we've talked about it with Christoph and Andy as well. We're not clear on how well the algorithm is actually moving between the different models. Is it possible to see that from the trace plots, for example?
Not the ones I've shown you, and not the ones that I have available to show you either. That would be a nice question to tackle directly, because a large part of why the Metropolis algorithms mix badly is because transitions between topologies are difficult. It's not too bad to write down Metropolis algorithms that mix over the branch lengths within one ranked topology. It's not too straightforward a thing to measure, because especially in the infinite sites case, where mixing is better in general, because so the algorithm is working on this space of ranked topologies, but actually a lot of those ranked topologies correspond to exactly the same coalescent tree. So, for instance, if you happen to have two sequences in your sample with exactly the same mutations, or let me put that another way, 3 samples with exactly the same mutations, then any topology in which those 3 merge together into a common ancestor in any order is, from the point of view of the target, exactly the same. So the question is, should you count those kinds of topology crossings as genuine topology crossings, for instance? If the answer is yes, then it would be quite easy to track how often you just cross a boundary. That wouldn't be hard, but it's not completely obvious that gives you a good sense of the mixing of the algorithm in terms of moving between genuinely different topologies.
Okay, thanks very much.
But yeah, one thing that I should say is that that's something that I also want to find a good way to measure that. So if you have any thoughts about it, then I would be very happy to hear about them.
Okay, thank you. So we've got one from Mark. Do you want to ask that one live, Mark? Okay, so Mark hasn't appeared. So he asks, would it help mixing to remove the restriction to ultrametric trees in phylogenetics branch length is proportional to the number of mutations?
Yeah, conceivably it could. One of my main motivations for this was to see if zig-zag could help with a particularly challenging model, which is why I didn't want to do that. That's sort of contrary to what one of the aims of the project was, but certainly in practice, that's something that one might want to do. That's one of the main reasons why I didn't use an off-the-shelf Metropolis-Hastings package in order to do the comparison with my zig-zag, was precisely because most of them do something like that, which changes the inference problem. So I mentioned earlier on in the talk that there was different embeddings of coalescent trees, and the most classical one, I guess, is Billera, Holmes, and Voigtman, which is exactly an embedding of trees that are not ultrametric, and that's what the HMC algorithm, for instance, uses. The tau space embedding that I used is specifically well suited for ultrametric trees, so you'd probably have to change the embedding that you're using to implement the algorithm, but apart from that, everything should push through, and I agree it would probably correspond to a computationally easier problem.
Okay, thanks. Is my microphone working now?
Yes.
Yes, you're on.
There's just tech.
Okay.
Thank you, Jere. That's a good answer. Yeah.
Great. So we have another Bristolian question from Christoph. So the topology exploration works because of the continuity at the boundary and the fact there is enough mass at the boundaries, right?
Yeah, it must be something like that. The boundary crossings are something that I've not explored as thoroughly, partly for the reason I gave in my answer to Anthony, and just because it's difficult to come up with good visualizations. But yeah, in principle, it must be something like that. I've certainly checked that the algorithm goes through a lot of transitions between ranked topologies. So, I mean, this is one of the reasons why coalescent posteriors typically end up being multimodal, is there's many different topologies that can explain any given data set, and it seems to be that there's enough mass between them that this momentum endowed by these velocities carries you through those boundaries quite nicely.
Okay, thanks. So I've just got a quick question. So in the implementation, so Jere's got one as well in a second. In the implementation you had of the Metropolis, so you're only using the SPR type moves rather than the things like moving neighbors. I mean, as the sample size increases where the Metropolis goes completely wrong, I'm guessing that you would need like these neighbor swapping moves to do anywhere near well. Yeah,
you're probably right. It probably ceases to be a fair comparison because these the boundary crossing moves of the zig-zag algorithm are precisely nearest neighbor interchanges. So those are the only moves in the topology space that the zig-zag does. So that would be a very fair criticism to level at the comparison. SPR is probably not the right one.
Okay. So Jere's question. Do you want to get live? Yep.
Yeah. So basically, I'm just trying to. So you when you showed your graphs, it seemed like increasing the number of sleeves is worse than increasing the mutation rate, right?
Yeah.
And does that happen precisely because, if I understand correctly, every time one of the intermediate times goes to 0, that's when you're changing the topology potentially.
Yeah.
And does it start working worse precisely because this becomes more often when you have more nodes?
I don't think that's too big an issue. It does slow down a little bit for that reason. But I think the main issue is the fact that the flip rates themselves are order n to evaluate. So the computational cost of evaluating a flip rate point-wise is linear in the number of leaves. And you need to do that typically many times in order to generate a transition.
So you're saying that even in a small number of leaves, I mean, how likely is it to change actually the topology, for example? I mean, how often does it actually happen? Is it like every move, almost every move, or is it?
No, not every move. I don't have a quantitative answer to that, except that, yeah, I've just sort of done runs where I've had the algorithm print what sort of outcome the next move had, and the topology changes happen sort of regularly. It's by no means every move, but it's not once every 200 moves either.
And that doesn't change when you increase the number of leaves, for example, because I would imagine that there should be more topology change proposals in the when you have more leaves. I mean, that sounds intuitive, at least to me.
Yeah, not proportionally though, because of this. So I had these velocities, which I've forgotten where it is in the presentation here. So when you add more leaves, you basically get more holding times, which means you have more velocities, but well, that's not worked. But let me see.
Okay.
The velocities themselves sort of scale like this, which means that, let me say it this way, if you were to take a tree for a sample of size n and add an n+1th sample, then you would add one extra holding time, and it would have a very short holding time but at the same time the velocity is scaled so that it sort of matches the scale of the holding time, and so yeah, you get more boundary crossings, but then you also get more events overall. So I'm not sure that the proportion changes very much.
Okay, thanks, and thanks for the talk.
My pleasure.
Okay, so that does seem to be the last question. So thank you very much again for the talk.
Thanks a lot.
Okay.
I'll stop sharing my screen